hustle.
You might have to, I think you push buttons. Yeah.
Okay, whatever. Hey guys, for those of you that, we'll just deal with it however it goes. For those of you that don't know me, my name's Adam. I am no longer a first year, I don't think. The Joe and Lux Labs, you guys can make that call, and I'll be doing the newsreel for today, which of course has to start out recognizing GeneMods has a podcast, and you should all totally go listen to it because it's awesome. So the first episode, SynBio in Space, which is the title with a lot of A's. It's like a Poly A or something, I don't know. Isaac and Jordan did a great job interviewing Professor Tolman-Urchak about some of the work that they're doing in that lab, kind of the space SynBio idea of how can we make recombinant proteins in space and what might be some of the useful ideas from that. And there's also at the end a game of wait, don't tell me, essentially, SynBio trivia, which is a lot of fun. I wanted to play this. I don't know if this is going to work, but we'll see if we have any volume.
Oh, yeah. These are the voyages of the micro Ecoli. It's a 5-year mission to colonize strange new worlds. We're going to talk about synthetic biology in space today with Dr.
So if that's a good enough hook for you, then go ahead and do that. I think the other highlight is learning that Isaac apparently doesn't know what bay means. So if you want to make fun of him for that too. Okay, so like Weston said, there's been a lot of conferences lately, so I'm not going to go too much into detail of things that happen in SB7 or C, because people that actually know things are going to do that instead. But a lot of the SB7 talks you can find either summaries or the full videos online. So just a couple quick things. The keynote was given by Randall Kirk, who's the CEO of Intrexon, which is a group that's been doing a lot of applications of SynBio for some interesting new GMO applications. So he was talking a lot about how they can make the salmon that are 3 times as big as the normal salmon, apples that don't brown over time. It's a really cool talk, and he has some choice language about gene drives and biofuels, which I thought was pretty interesting. There was an entire session at the conference devoted to biodiversity, which I think is an issue that don't necessarily hear talked about that much with synthetic biology, but there are people that are doing things. I think a lot of us know about the work that the Church Lab is doing.
We're going to actually live stream this presentation, which is kind of cool. So we're trying that out. People who are not here, I guess mostly if you're here, just raise your hand and ask questions. But people can also actually live stream in questions as well. And so Joe is the monitor for that. And so if someone from the interwebs has a question, then they'll have Joe, they'll conview it through Joe. So anyway,
This is now the
it's pretty exciting, right? You're on the web. Okay. With that, let's thank Adam.
Good hustle.
You might have to, I think you push buttons. Yeah.
Okay, whatever. Hey guys, for those of you that, we'll just deal with it however it goes. For those of you that don't know me, my name's Adam. I am no longer a first year, I don't think. The Joe and Lux Labs, you guys can make that call, and I'll be doing the newsreel for today, which of course has to start out recognizing GeneMods has a podcast, and you should all totally go listen to it because it's awesome. So the first episode, SynBio in Space, which is the title with a lot of A's. It's like a Poly A or something, I don't know. Isaac and Jordan did a great job interviewing Professor Tolman-Urchak about some of the work that they're doing in that lab, kind of the space SynBio idea of how can we make recombinant proteins in space and what might be some of the useful ideas from that. And there's also at the end a game of wait, don't tell me, essentially, SynBio trivia, which is a lot of fun. I wanted to play this. I don't know if this is going to work, but we'll see if we have any volume.
Oh, yeah. These are the voyages of the micro Ecoli. It's a 5-year mission to colonize strange new worlds. We're going to talk about synthetic biology in space today with Dr.
So if that's a good enough hook for you, then go ahead and do that. I think the other highlight is learning that Isaac apparently doesn't know what bay means. So if you want to make fun of him for that too. Okay, so like Weston said, there's been a lot of conferences lately, so I'm not going to go too much into detail of things that happen in SB7 or C, because people that actually know things are going to do that instead. But a lot of the SB7 talks you can find either summaries or the full videos online. So just a couple quick things. The keynote was given by Randall Kirk, who's the CEO of Intrexon, which is a group that's been doing a lot of applications of SynBio for some interesting new GMO applications. So he was talking a lot about how they can make the salmon that are 3 times as big as the normal salmon, apples that don't brown over time. It's a really cool talk, and he has some choice language about gene drives and biofuels, which I thought was pretty interesting. There was an entire session at the conference devoted to biodiversity, which I think is an issue that don't necessarily hear talked about that much with synthetic biology, but there are people that are doing things. I think a lot of us know about the work that the Church Lab is doing and trying to bring back woolly mammoths. There's also some interesting ideas, for instance, Ryan Phelan, who's talking about how they can engineer coral to be more resistant to bleaching, prevent environmental loss in that way. And then probably the big news that came out of this, there was a note that was sent out to the listservs that Twist Biosciences is partnering with BioBricks to provide 10,000 genes to the public free of charge. You're going to be able to vote on these somehow online. I'm sure Isaac's going to talk a lot more about this later, so I'll let them have the honors. Some more community news. So SynBio published this interesting interview with Julie Legault, who is the founder of Amino Labs. Amino Labs is kind of, it's a company that's devoted to spreading SynBio as a form of outreach to younger kids to try to get them involved in STEM especially. So this is a way of using microbes to make fragrances, colors. She's actually from the MIT Media Lab. That's where she got her start, and then thought that synthetic biology was a really cool way of sort of bringing this science to younger people. And you can actually buy one of these kits where you can make the different fragrances and colors. Community news on the CRISPR side of things, just to make things even more complicated, China sided with the Doudna side on the patent for CRISPR, just like the EU now. So the patent in the EU goes against what's currently the state in the US. So we have a picture of Doudna smiling, and this is the angriest picture I found of something online. It turns out if you Google like Feng Zheng frowning, like you just don't get a lot of great things. Also in Doudna news, she's got a book out. It's called The Cracking Creation, and if you are more perceptive than me, then it will take you less than like an hour to figure out that like the letters A, C, T, and G are the ones that are yellow in that title. It's essentially a memoir of the CRISPR saga. The reviews online have been sort of mostly positive, I would say. I haven't gotten the chance to read it or anything yet, but if you want to hear her words about what role she and others had to play in this discovery, then go ahead and check it out. More on Twist. So Microsoft is spent a bunch more money buying more genes from Twist in sort of their goal to encode all of the world's data into DNA. So this was another $10 million arrangement adding on to all the DNA that they bought last year. And I think Catherine talked about some of the work that had happened a couple of GeneMods ago on that front. IDT is also in the news this week. So they launched this new grant program that's specifically devoted to SynBio startup companies. So you can apply with what you think your SynBio startup will do, and they will reward you in G-blocks. So I did a shout out here for our good friend James, who's leaving the Lux Lab later this week. He's always saying, always be cloning. So first place you get 100,000, second place 50,000, third place you're fired. ABCs, right? Okay. On the other side of things, so Bolt Threads, which is one of the companies that's really been involved in getting the spider silk stuff up and running, the yeast brewed or micro brewed spider silk that can potentially go into clothing. They signed this deal with Patagonia, so they're actually making like winter clothing out of this stuff. I was curious what the state is of this. You can actually buy one of these ties, and it sort of looks like spider silk, I guess, but it could like probably pass, and it's only $300. So, like, that's more than I would spend on a tie, as you can tell on the slot, but like, you guys could do it. On the news, on the research side of things, there's an interesting perspective piece in Nature Biotech, primarily focusing on outreach and introducing younger students in high school to STEM, synthetic biology, so programs like iGEM, and then they also talk a lot about the Canadian equivalents of iGEM, which are a thing apparently. They look at a bunch of surveys from the people that go to the iGEM Jamborees. They also have this kind of interesting bar graph I found of the participation as it's changed over time. So in the blue there is the college programs doing iGEM, and in green is high school, which has come up a lot later. What I thought was interesting actually is how the balance has shifted in recent years toward more Asian programs that are participating in this. So it's really becoming more of an international scope type of a thing. More research. So the Arkin Lab published a sort of interesting paper with potential applications for metabolic engineering. This programming mRNA decay, which takes advantage of a native this MASF endonuclease, which is targeted to specific regions in native mRNAs and chops them up. And what the Arkin Lab found was that if you delete you recode the target proteins that you want in enzymes that are participating in your metabolic pathway, you can reduce and shift some of those endonucleases away from degrading the proteins that you want and potentially improve titers. But what's cool is that it's actually a stable system if you do this. So they do a lot of the control theory behind the scenes to show that this has a feedback mechanism in place to get very stable regulation. The Baker Lab kind of just pulled another Baker Lab. So this time, probably whoever went to seed is going to talk a little bit about this. But probably the most interesting thing they have out here is this computational design of a trimeric binding protein to hemagglutinin, which is a cell surface receptor on cells that are infected by flu virus. So one challenge that people always run into when you're, for instance, making a flu vaccine is they make these like trivalent vaccines because we don't really know what the binding pockets are going to be every year. So what they did is they actually found that they can engineer these proteins de novo, as the Baker Lab is wont to do, and they can get really, really good. If you look on the right there, the red line is when they use these proteins as a prophylactic, and it really helps the mice survive the flu better. An interesting paper published more in metabolic engineering. This was actually done in collaboration with ExxonMobil, engineering algae to be better lipid producers. So they found that if they just delete this one transcriptional regulator from the gene, they can turn the cells that look like this into cells that look like this, where 60% of the cell is actually making lipids. That's comparable to the sort of yields you see in urea, which is sort of the workhorse for making industrial lipids, but this is done in algae, so there's a lot of potential benefit that way. CRISPR news. For those of you that haven't seen this, this has been sort of a mess. It all sort of started out this last week in May. This lab in Iowa, I think it's actually more of a hospital, the Mahajan is the last author in this paper, publishes unexpected mutations after CRISPR-Cas9 editing in vivo. So they did a bunch of germline mutations to mice using CRISPR. Things seemed to work, and then they did whole genome sequencing and found that there were like dozens and dozens and dozens of off-target effects, indels, deleterious mutations all over the place, not where they were trying to mutate, and also not where computational design algorithms would predict the most likely place that they would get off-target effects. So this was quite bad, and once this paper hit, stock price of Editas dropped precipitously. All the CRISPR biotech companies started dropping precipitously. People didn't really quite know what to do with it. I guess it's recovered a little bit since then, possibly on the strength of the resulting paper that got published on bioRxiv just about 2 weeks later, where this is from Editas saying basically you guys screwed this up. This is a really fun paper to read, honestly. They basically said, you guys suck at what you do, we don't believe your results. There were definitely some suspicious things in the original study, so they only used 3 total mice. They had 2 treated mice and 1 control mouse, and the 2 treated mice were like relatives of each other. They were siblings, where the other one wasn't. They said the sequence depth wasn't good enough to get these to draw the conclusions that they had, and so on and so on. You can sort of see George Church laughing at them. Again, if you Google George Church laughing, that was the best I came up with. They published a follow-up to the rebuttal now, and that's also on bioRxiv, where they're essentially saying, yeah, it wasn't necessarily the best experiment, but we stand by our data. We think this is real and something that people need to be aware of. I love the end of it. Overall, we are optimistic that some form of CRISPR therapy will be successfully engineered to treat blindness. So So we're all on the same page there. And then this is George Church with black eye. It turns out if you Google George Church, this is like the 5th image that comes up. So, I saw that picture and I was like, this is going in the presentation somehow. All right, back on actual science. So the Silver Lab published a really cool paper talking about more of this gut microbiome stuff, engineering E. coli that can actually function as sensors inside of the mammalian gut. So what these bacteria are is they're able to sense a marker of inflammation called tetrathionate, which will switch on a genetic circuit, essentially a toggle. And what they find is that mapping the genome of these organisms after a month, they're able to maintain that toggle switch being active, which is really cool because you can potentially use this as a diagnostic tool or potentially even to deliver a therapeutic. A bunch of new cloning things happened. If you're interested in cloning, which I'm sure you all are, I thought the coolest one was actually this paper from NAR, which is essentially a new way of doing non-enzymatic DNA assembly, taking advantage of a lot of the techniques that people do when they do Gibson, but rather than sort of creating nicks using exonucleases and then filling them in, what they do is they create like 2 sets of overhangs for every for every construct that you're trying to assemble, and then they denature them and then they re-anneal them so that they can get the fragments to overlap like one after the other after the other. They finally get really great fidelity, comparable to Gibson, also really great transfer efficiency for the assembly, and potentially could be a lot cheaper because you don't have to pay for all the enzymes that go into a Gibson assembly. And you had a bunch of papers out, so Colin in the Lux lab got this paper published in SynBio on a split broccoli, which is a great way that people could potentially measure RNA-RNA interactions without the need of stupid proteins. Sorry. Tough crowd. A lot of people in the Jewett lab here, Weston, other people I'm sure somewhere out here. This paper on a cell-free platform for doing glycosylation just came out. I think it's a good preface to a lot of other work that's going on in that lab. Tolman Ursek lab published also in SynBio about some new improvements that you can make microcompartments to improve the diffusion into the spaces, and potentially that would be really useful for pathway optimization, metabolic engineering type things. I caught this, it looks like a preface to a review that's got Devin from the Leonard lab on it on exosome delivery to breast cancer cells, so looking forward to seeing more in that space as well. I think that's all I have. Any questions? No one from online, please.
Not yet.
Please. Please don't do it. I can't handle it.